Why cancer still wins.
Humans can build supercomputers and send spacecraft into deep space. So why are we still unable to defeat a handful of tiny cells growing inside our own bodies? It is one of the strangest paradoxes of the modern age. We can send probes millions of kilometers from Earth, yet when it comes to cancer, there is still no simple answer. To understand why, we first have to understand how cancer actually works, because this enemy is far more complicated than it appears. One of the biggest mistakes we make is thinking of cancer as a single monster. We use one word to describe it, which makes it easy to imagine that all we need is one powerful drug to destroy it. But that single name hides an enormous amount of complexity. In reality, we are not fighting one enemy at all. Cancer is more like a massive army made up of hundreds of different diseases pretending to be one. There are more than 200 types of cancer, classified by where they begin in the body and the type of cell they develop from. Skin cancer, lung cancer, and blood cancer are not simply the same disease appearing in different places. Each can follow its own rules, grow at a different speed, and attack the body in a different way. Finding one universal cure would be like trying to design a single key that could open hundreds of completely different locks. Even two people diagnosed with the same lung cancer can have tumors as different as fingerprints. On the surface, the diagnosis may sound identical, but deep inside the cells, the genetic errors and molecular features can be very different. One tumor may depend on a particular growth signal, while another survives through an entirely different pathway. That is why a drug can work extremely well for one patient and barely work at all for another. It can begin when something goes wrong in the DNA instructions that control how a cell behaves, allowing the cell to ignore signals that normally limit its growth. Healthy cells follow a carefully controlled cycle. They grow when needed, divide when permitted, and eventually age or remove themselves when they are damaged or no longer useful. Those rules help protect the body from chaos. Cancer cells break them. They continue multiplying even when the body has no need for more cells, gradually crowding out healthy tissue. Imagine a car with the accelerator permanently stuck to the floor. That is one way to picture what happens inside many cancer cells. Genes that promote growth can become abnormally active, like an engine being pushed at full power without stopping. Instead of receiving a go signal only when the body needs new cells, the cancer cell keeps receiving or generating that message. It divides again and again, producing more abnormal cells and continuing forward without slowing down.
When the brakes fail.
But a car also has brakes, and this is where the problem becomes even worse. A cell has its own emergency braking systems, including genes known as tumor suppressors, and those systems can also become damaged. Normally, when a cell detects serious problems, these genes can tell it to stop dividing or trigger other protective responses. Cancer can disable those safeguards. When the accelerator is stuck and the brakes no longer work properly, abnormal cells are free to keep multiplying and invade the healthy tissue around them. Most normal cells also have a biological limit on how many times they can divide, but many cancer cells find ways to push past that limit and become almost immortal. With each division, normal cells gradually move closer to the point where they can no longer keep dividing. Many cancer cells, however, activate an enzyme called telomerase, which helps maintain structures associated with continued cell division. This allows them to avoid some of the normal aging processes that help keep cell numbers under control. A cell that refuses to stop growing has now also found a way to keep going for far longer than it should. Cancer cells can also become masters of disguise, tricking the immune system into treating them more like harmless members of the body than dangerous intruders. Normally, Immune cells patrol tissues looking for signs that something is wrong, but cancer cells can display signals that suppress or weaken the immune response, almost like showing a convincing security badge to a guard. Immune cells may fail to attack or may become less effective around the tumor. That gives cancer more time to establish itself and continue growing inside the body, and any growing stronghold needs supplies. Tumors can release signals that encourage the body to build new blood vessels around them, giving them greater access to oxygen and nutrients. In other words, cancer does more than break the rules of growth, disable cellular breaks, extend its ability to survive, and evade the immune system. It can also help create its own supply network. That steady flow of resources allows a tumor to keep expanding and makes it even harder to eliminate. Cancer cells do not always stay where they began. Some learn how to break away, enter the bloodstream or lymphatic system, and establish new colonies in distant parts of the body. This process is called metastasis. A single cancer cell can leave its original tumor, survive the journey through the body, reach another organ, and begin forming a new tumor there. That is why finding and eliminating countless metastatic cells scattered throughout the body is one of the greatest challenges doctors face. But one of cancer's most remarkable abilities is its capacity to change in ways that help it escape treatment. When a therapy targets a particular biological feature, some cancer cells can alter their state or behavior. A drug that once recognized and attacked them effectively may suddenly become far less useful, almost like chasing someone who keeps changing their appearance. This flexibility gives tumors a powerful ability to adapt and can allow them to stay one step ahead of treatment. A cancer cell may begin with the characteristics of a lung cell, yet shift into a different cellular state simply to survive treatment. This ability is known as phenotypic plasticity, and it can help cancer cells escape therapies aimed at very specific targets. Cancer cells are not always locked into one fixed identity. When their environment becomes hostile, some can change how they behave and which biological programs they use. In that sense, they are shape-shifters, capable of changing themselves just enough to survive. A tumor full of survivors. The chaos does not stop there. Even a single tumor can resemble a crowded, disorganized city filled with many different kinds of inhabitants. Cells near the center may behave differently from those near the edges, with different weaknesses, growth rates, and levels of resistance. A treatment might destroy most of the tumor while leaving behind a small group with very different characteristics. And if even that small group survives, the battle is not over. This is where natural selection begins to play out inside the body itself. Treatment kills the cancer cells that are most vulnerable, but cells that happen to be more resistant are more likely to survive. Those survivors continue multiplying and pass on the traits that help them withstand treatment. Over time, the tumor population can become dominated by cells that the original drug can no longer control. A treatment that once worked extremely well may eventually lose much of its effectiveness. In that sense, cancer is evolving inside the body much like populations evolve in nature, only on a much shorter timescale. New genetic changes can continue to appear, and under the pressure of treatment, any change that helps a cell survive can give it an advantage. The cruel irony is that each round of therapy can act like a test. Most cancer cells fail, but the ones that survive may be the hardest ones to kill. That is one reason cancer treatment often has to change as the disease changes. Even when a drug can still kill the cancer, it first has to reach the right cells. Some tumors develop dense surroundings that act almost like defensive walls. Thick networks of proteins, connective tissue, and supporting cells can create a complicated tumor microenvironment around them. These structures can make it difficult for drugs to penetrate deeply into the tumor. So even when medicine reaches the tumor itself, it may still fail to reach every cancer cell hidden inside. Brain tumors create an even more difficult problem because the brain is protected by an unusually strict security system called the blood-brain barrier. Its job is to prevent many potentially harmful substances in the bloodstream from entering brain tissue. Unfortunately, that same protection can also keep cancer drugs out. Many treatments have difficulty crossing this barrier in useful amounts. Doctors, therefore, face a delicate challenge: getting enough medicine through this biological security gate to attack the tumor without damaging healthy brain tissue. Even more surprising, some cancer cells can survive treatment without acquiring a special new mutation at all. Sometimes survival comes from chance and from ordinary biological variation between cells. A few cells may happen to be in a quieter, less active state when treatment arrives, making them less vulnerable to drugs that work best against rapidly growing cells. Once treatment ends, those cells can become active again. These temporary survivors may later provide the seeds from which the disease returns. Cancer that sleeps for years. And some cancer cells can remain hidden for far longer. They can behave almost like sleeper agents, entering a dormant state and remaining quiet for years before becoming active again. Because they are dividing very slowly or not at all, treatments that mainly attack growing cells may have difficulty eliminating them. The body may appear to be free of disease, while a small number of dormant cells remain. In some cancers, those cells can become active again, even 10 or 20 years later, allowing the disease to return long after it seemed to be gone. Developing a new cancer drug is an incredibly difficult process, and only about 5 out of every 100 candidates ultimately make it to approval. Behind every treatment that reaches a hospital are countless promising ideas that failed somewhere along the way. Cancer drug development has one of the highest failure rates in medicine. Many compounds can destroy cancer cells beautifully in a laboratory dish, only to behave very differently once they enter the complexity of the human body. Testing drugs in mice is extremely valuable, but a mouse is not a human, and cancer does not behave exactly the same way in both. Animal models allow scientists to study potential treatments before exposing people to them, yet the mouse immune system and tumor environment differ from ours in important ways. As a result, a drug can look remarkably successful in mice and then fail when it reaches clinical trials. That gap between the laboratory and the hospital remains one of the hardest obstacles in cancer research. Even after scientists understand the mutations hidden in a cancer cell's DNA, another layer of complexity remains. Cancer can switch genes on and off without changing the DNA sequence itself, almost like using a hidden control panel. By changing how genetic instructions are read, cancer cells can adapt to stressful conditions while keeping the underlying DNA largely unchanged. The genetic code may look the same, but the cell can behave very differently. This makes treatment harder because there is not always one permanent mutation that doctors can simply target. Even more surprisingly, the bacteria living inside our gut may influence how cancer responds to treatment. The gut microbiome constantly interacts with the immune system. Some microbes may strengthen certain immune responses, while others can reduce the effectiveness of treatment or create conditions that help tumors survive. Researchers are now investigating whether changing these microbial communities could improve the chances of treatment success. A battle that seems to take place entirely inside a tumor may actually involve an entire ecosystem living inside the intestine. New ways to fight back. Because cancer is so complicated, treatment is becoming increasingly precise. Scientists have developed targeted therapies that work almost like keys designed to fit very specific locks. Instead of attacking every rapidly dividing cell, These drugs search for particular molecular abnormalities that cancer cells depend on. This can spare more healthy tissue and reduce some of the damage caused by less selective treatments. But there is an important limitation. The key only works if the tumor actually contains the lock it was designed to open. We are also learning how to turn our own immune cells into highly specialized cancer hunters. CAR T therapy begins by collecting T cells from a patient's blood, genetically modifying them so they can recognize a particular target, and then returning them to the body. Once inside, these engineered cells can identify and attack certain cancer cells far more effectively than before. For some cancers, this approach has already shown how powerful a patient's own immune system can become when it is given the right instructions. Another strategy sounds almost like science fiction, using extremely small delivery systems to carry drugs more directly toward tumors. Nanoparticles can be designed to transport medicines through the body and help concentrate them in or around cancerous tissue. Researchers hope these systems can improve how deeply drugs penetrate tumors while reducing exposure to healthy tissue. The goal is simple: deliver more of the treatment where it is needed and less where it is not. But perhaps defeating cancer does not always mean eliminating every last cancer cell. In some situations, a more realistic goal may be to turn cancer into a long-term manageable disease. Instead of trying to destroy every cell at once, doctors may focus on keeping a tumor stable, preventing it from spreading, and stopping it from causing serious harm. Much like other chronic illnesses, some cancers can sometimes be controlled for long periods with ongoing treatment. In that case, victory means preventing cancer from taking control of a person's life. The future may involve watching cancer far more closely and changing treatment as soon as the disease begins to adapt. Highly sensitive blood tests are being studied as a way to detect tiny traces of tumor material and reveal how a cancer is changing over time. If signs of treatment resistance can be detected early enough, doctors may be able to adjust therapy before a resistant population has time to take over. Cancer treatment could become an intelligent pursuit, constantly responding to the disease instead of relying on one strategy from beginning to end. The struggle against cancer reminds us just how astonishingly complex the human body really is, and how much persistence matters in understanding it. Every mystery scientists solve teaches us something new about life, disease, and the limits of our own biology. We still do not have one universal cure for the hundreds of diseases we collectively call cancer, but knowledge, technology, and human care continue to help more people survive and live longer. The final victory may not come from one miraculous pill. It may come from thousands of smaller breakthroughs, each building on the one before it.